Okay. Welcome to the afternoon session. The first talk will be a keynote speech. We have 3 keynote speeches. We had planned in the beginning to have one every day, but then the commitments of one of the speakers forced us to put 2 keynote speeches in the same day. But this is an advantage from the viewpoint of the conference, because Friday afternoon will be particularly interesting, so people will be discouraged from leaving earlier. This was one thing. Okay, now the first keynote speech by Joana Xavier. I met her perhaps 4 years ago, something like that, in Santa Fe, when there was the Stuart Kaufman 80th birthday. He invited a number of people. We had a wonderful time and discussing in a very open-minded way. And I don't want to anticipate much about what Joana will say. Stu is very, very fond of her work, so it's one thing which I think we should take seriously. Joana is a computational biologist and a bioengineer at a new company which is called RAFs-Labs, and she has got a PhD in chemical and biological engineering from the EMBL in Germany and the University of Minho in Portugal. She also worked at Argonne National Laboratory. She has been working in very smart places. She has been a postdoc in Düsseldorf and also in a University College London. She has worked with very smart people. She's also, I was forgetting, she's the founder of the International Origin of Life Early Career Network, which is something which I think it's interesting to see what's happening. And so, fine, I don't anticipate anything about her topic. She will do it.
Thank you. Thank you, Roberto. I'm so glad to be here. It's really an honor to give a keynote in such an interesting meeting, so diverse. As Roberto said, I met him in Stu Kaufman's birthday conference, which was one of the most interesting scientific events I had the pleasure to attend. And I'm really glad to see you again. That was 5 years ago. Wow, time flies. So today I'm going to talk about autocatalysis in general and a bit in depth, and the origins of life on Earth, which is my passion since a long time ago. So I want to start with acknowledgements. I think it's a bit of a mistake to leave them to the end. I am the sum of the parts that made me, and all of these people, in particular for this talk, are very, very relevant. Josh and Steve are the co-founders of RAFs-Labs, where I am employee number 1. We just started this year. We are very interested in the origin of life and artificial life, and we build AI models for the chemistry of life. We are named after Margaret Dayhoff. If you don't know her, you should. She's one of the under-recognized women in science. She's the founder of bioinformatics, in my view, at least in protein bioinformatics. So we want to honor her with this project. Until very recently, I was in Nick Lane's lab in UCL. So I'm very grateful to Nick as well. He's a very inspiring mentor. You should also read his books if you can. And thanks to all my funders. Yeah, if you're interested in origins, go for the vital question. If you're interested in chemistry a bit deeper, go for the latest one, the transformer. Yeah. So I'm going to start talking about Earth's life and its universal properties. The reason why I want to do this is because for this audience, I think you guys like to think of abstractions and models, and I do too, very much. I was just telling Roberto, the sweet space for me is right in between the world of abstraction and a lot of data. And that's a bit of a daunting place to want to be in, but that's where I try to position myself. And I want to start with the data, the evidence that we have on what life is. It's a big debate, but I'm going to focus on what we see, the evidence, the life that exists on Earth, right? And a bit of its universal properties, which exist, and we should not ignore them. So this is a quote from Marie Curie that inspires me very deeply as well. I've wondered about life since I remember I'm very lucky to be doing what I want to do. When I was in college, I was already thinking about the origins of life. It was not an accident that I ended up studying life and its origins. There are many definitions, and I'm not going to go deep or challenge too much about what life is, but I'm going to tell you at least one that life is not. Life is not order. And a lot of you may still be enticed with that concept, but there's a lot of order in the universe outside of the living world. This is an example. It's a relatively complex molecule that you find in the mineral world. I took this picture in the Natural History Museum in London, where I live. It's the microstructure of a beryl crystal. And if you show it to early students, they will usually mistake them with a protein or even DNA if they don't look too closely. But the fact is, order exists in the universe before life. And this type of order is actually very important for the origins of life, as in my perspective, as I'll try to tell you. So I'm a bioengineer by training, which I consider myself very lucky because I learned a lot of different things, a lot of mathematics, a lot of bios, modeling, simulation, chemistry, physics, even some economics and computation. But when I was in college, all the other engineers were building and taking their stuff apart, and I was like, I can't do that. I can just wonder at these things and think how the heck did the universe made them. And it became an obsession. And it's an obsession that is with me for 15 years now. So it took me to the root of the great tree, of course, which is still a big mystery, but I believe we're making some progress recently as a community. So I'm going to tell you a bit about my own work in this talk, but also a global perspective, very driven by my interaction with early career researchers in the origins of life. So prokaryotes, they are at the base of the tree. They are as far as we can go in terms of simplicity, but they are exquisitely complex, as you know. And the numbers are just astonishing. There are more than stars in the universe. If you take 1 gram of dental plaque, that we have probably more here in this group, we shouldn't, but there are more bacteria in 1 gram of dental plaque than the number of humans that have ever lived. So the picture we have of life is driven by what we could observe so far. We didn't know this. We didn't know them. We're starting to know them now. And we need to pay a lot of attention to them if we want to think about the origins of life, because if we're talking about the origins of life that we see, that we know, we're actually talking about the origins of prokaryotes. So this is a commonly shown picture when you teach kids, students about prokaryotes. It's wrong, it's bad, don't do it, please. This is still in the internet, but this is not what a prokaryote is. I wouldn't blame people too much because the real picture of what a bacteria is, which is much denser, the cytoplasm is much more like a gel, it's not a bag of water. I got this picture on my PhD cake in 2011. So this is a drawing by David Goodsell to scale of mycoplasma. So it's recent. Okay, I admit it's recent. But we have it now. We have full pictures of what these things are, and we can study them. And we're doing even better now. This 3D model, if I can play it, was just published last year by a very bright student of David Goodsell in California. And for me, this is something that we cannot ignore if we start to think about the origins of life. And it has to be at the front and center of how we think about the question. As you can see, it's not a bag like the first thing we saw. It's a very dense world of protein and nucleic acids and small molecules. So when we talk about what is universal in life, here you go. These are universal things that all prokaryotes have, okay? You have the boundary maker, which is a lipid synthase. You have what I call the grand translators, which are ribosomes and ribosomal proteins. You have the transcriber, RNA polymerase, the copier, DNA polymerase, and what I like to call Maxwell gnomes, which I think will have a much more impact in how we think about the origin of life in the next decades. Proteins in the membrane. We know much less about them because they're much harder to crystallize, but they kind of establish what is self, what is life from what is not. So they are extremely important. So universally essential, take them into account. There are universally essential principles of organization in life, the way proteins are arranged. This is what the authors call the periodic table of protein complexes. So you find these principles of organization in all of life. So take them into account. Also, we heard this morning about my favorite part of what is universally essential for life, metabolism, metabolic networks, all the biochemistry that sustains homeostasis, an important concept that I'm going to talk about a bit later, growth, replication. Metabolism is actually the majority of the genes that cells express, together with genetic information processing. There are millions of reactions happening every second in a single cell. So a lot of complexity for us to detangle, but we're starting to do it. There are core essential chemical reactions independent of genome shuffling and LGT in the prokaryotic world. And there are no universally shared genes. You may know this. But reactions in chemistry are universally distributed, as you'll see in some of my results. Also, metabolism is collectively autocatalytic, which will take me to what you guys are all interested in, I think, autocatalysis. But first, I want to tell you just a little bit of results of my work trying to find universally essential cofactors in prokaryotes. These are catalysts, small molecules that all of them use to accelerate themselves, their production. This was a work that integrated simulations from 70 genome-scale metabolic models. We heard a bit about that before, and a lot of literature and a lot of data. And so we established these 7 classes of cofactors that are essential. They seem to trace LUCA because they're essential for all prokaryotic life that we know. They are corroborated in another study of LUCA in 2016 in Nature Microbiology. And later, they were integrated in community modeling standards. So this is a good example of how when you really study the principles of life with data, it will apply to its origins, but it will also apply in the way you think about it and you model it and you make predictions. So universally essential organic cofactors. So the picture that we have of the origin of life right now is pretty bad, as you can see by my bad drawing in the middle. But what we know is that there has to be some kind of connection between a geochemistry in the beginning of Earth and the biochemistry we observe today. The middle is a black hole. We have quite some knowledge about LUCA that people are kind of agreeing now. We have a lot of ideas about the complex networks and the complexity before, but then there's that gap there. How do we get verticality before genetic encoding? FUCA, the first universal common ancestor, also called EDA, the initial Darwinian ancestor, big mystery. So this is where I did some work on autocatalysis that proved interesting. And let's go to autocatalysis then. I'm going to talk a bit about some shoulders where we stand, at least where I stand. You may be interested in more, and I would love to discuss them all. About RAFs, which is the particular framework that I used to get these biochemical results. And then I think I'm going to talk about hydrothermal flow later. So let's go. Autocatalysis is a really interesting concept because it lets us detangle the self-referential paradox. Everyone is fascinated with the self-referential paradox. Louis Pasteur kind of realized it when he showed us that all life comes from life. But then how do we solve the origin of life? The genetic code is also self-referential. Genetic code makes proteins, proteins make genes. It's a $10 million question that I'll talk about in the end. Yeah, so the transformation of a cell to cells is autocatalytic conceptually. It's self-referential. In the most simple form, autocatalysis is a reaction. There you have two reagents in green that produce a product in black. That catalyzes, the reaction is the square. That catalyzes its own formation by being a catalyst to the reaction that produces it. So very simple concept in this level, and then it becomes more complicated. Last year, I wrote a paper with Klaus and Zen. We met via Olin, so you can see that Olin, the Origin of Life World Career Network, is starting to have an impact in the field. We tried to detangle a bit of this concept because there is a lot of discussion today because everyone is so excited with autocatalysis. I'm invited to give talks all the time, which is amazing, but then there's a lot of debate. So we went back to the original paper, 1890, by Wilhelm Haushalt, and we translated a bit of it. Actually, Klaus did most of it. I speak a bit of German, but not that good. So we translated it so you can have a notion of how this concept was proposed. I like how people used to write then. The question arose in my mind whether a substance subject to a slow reaction can have a catalytic effect on itself. And something that we talk about in this paper is how Oswald was not only talking about products that accelerate their own formation. He also considered the possibility of a reagent being also a catalyst in its degradation. And I'm not going to go very deeply here, but if you're interested in autocatalysis in this very fundamental level, please read the paper, because if you're looking for autocatalytic networks, like I am, and you take this concept of recessive autocatalysis, as we call it, you can then reverse it, because in theory all reactions are reversible, and you can try to find autocatalysis with much more possibilities. So, note. Autocatalytic reaction, easy to understand. Autocatalytic network or system, things start to get complicated here. So, there are some people that were very influential in the way that I think about autocatalytic systems. Two of them are here, Maturana and Varela. Francisco Varela, very inspiring person in many other ways, but scientifically this concept was not well received when it came out, but recently I think it's gaining the traction that it should. But how is it different, autopoiesis from autocatalysis? The way that Varela defined it later in his life was a network of processes or molecules that interact in such a way as to produce the very network that produced them, and that determine its boundary. Kind of a wordy definition, right? So the very difference between autocatalytic processes and autopoietic processes is that the latter, autopoietic processes, actually determine their topology. This means they are involved in the creation of their boundary. And this is not something that an autocatalytic system, at least as traditionally defined, does, because there are conditions around it that enable it, but those conditions are independent from the autocatalytic system. The autopoietic system is responsible for the whole thing. So the topology, as I said, is determined by a container, and this is something that I find really interesting, and I would love to debate it with you, but in their view, and in mine too, reproduction must necessarily have arisen as a complication of autopoiesis during autopoiesis. So the origin of reproduction, as opposed to the mainstream thinking, is secondary to and independent from the origin of the living organization. So here is a quote that I think really captures the idea of autopoiesis is, for the origin of an autopoietic system, what matters is the conditions that must be satisfied. This problem is not a chemical one. of what molecules take part, but one of relations between the units. I have a little bit of a quibble with this because I think those relations are dependent on the properties of the molecules very deeply. Especially, I can't see life without carbon and carbon orbitals, but that's another question. But yeah, a lot of food for thought. Yes, see also Tibor Ganti, because it's a very related concept. Both papers came in the same year. Tibor Ganti Schematon, nice translation by Urs Satmari. Very, very similar, but I don't have time to talk about all of them. Another of the pillars where I stand is Prigogine, and Prigogine also calls our attention to the environment, the container, and says, look at the energy. Because there are fluctuations in all systems that generate structures, and then there are bifurcations. We were talking about this at lunch a bit, a critical point where a system has a qualitative change. It's almost the moment of emergence. And both lead to development of lifelike complexity in Prigogine's view. So in open systems, which living systems are, and physicists don't love open systems, you gotta start thinking about them a little more, energy input leads to the creation of order. So life does not oppose the second law. Leaving all the problems with order aside, the interplay between determinism and chance, in the view of Prigogine, leads to chemical evolution. So in his book, Order Out of Chaos, he talks about autocatalysis a lot. It's not the central thesis that he has. His thesis is mostly about dissipation and energy. But you can read Order Out of Chaos. It's a really good book. So this quote, the interaction of a system with the outside world is the starting point for the formation of dynamic structures. So again, environment. Finally, before my mentor Stu Kaufman, I want to mention Freeman Dyson. And I want to mention him because he did write an interesting model of a self-sustaining system. It was not anything too out of the blue, but there's a quote in his book, Origins of Life, in 1985, that really got my attention, and I hope it gets yours too. He's talking about the Spiegelman experiments with RNA replication, Spiegelman and Eigen experiments. One of them put a virus in there, the other didn't. So he says, these experiments show how simple the phenomenon of replication is compared with the phenomenon of homeostasis. I am conjecturing that the minimum population size required for homeostasis would be about 100 times larger than that of reproduction. So he suggests that experiments similar to those of Spiegelman and Eigen would be the way forward in Origins of Life, just focusing on homeostasis. And I think so too. I really think so too. Rosen, I have to mention Rosen. Life Itself is a transformational book. It's such a hard book. Has anyone read Life Itself? Oh, it's so hard to read the first half, but once you get through it, it's like, my life has changed. His theory of autocatalysis is very much, it's as everyone saying, it's about the relation, the organizations of relations. He talks about MR systems, that's the name he chose, metabolism and repair, the minimal capabilities of a material system to be alive. He does not mention autocatalysis, no one knows why, but you can see here, this is an autocatalytic system, the minimal you can have, where you have a product that is catalyzing its own formation via the nutrients that are available. It's also a RAF, which I'm going to tell you later about. So Rosen says every MR system is a little theory of fabrication. He also opposes very much the machine metaphor, which I really support that we should oppose. This book, really, if you like mathematics, physics, abstractions applied to life, that's the one. Okay, we're to my first idol, then mentor, now friend, Stu Kaufman. We've been working together for the past 6 years. It's quite astonishing. When I was in college, I never dreamed that I could meet him, and then through hard work and tenacity, I got to meet him, and we became good friends now. That is not Sue's birthday, or I'm so sorry about that. So there's a reason why I was finishing my talk this morning, and that is the hotel doesn't have internet. So next time, other hotel. And yeah, it was a bit rushed. I'm so sorry. This is terrible. I think he would laugh, yeah. So Stu tells us to look up the Kantian hole. He tells us replication is not a property of it's a property of a complex dynamic system, not of a single molecule. Again, look at the system. In such a system, no molecule needs to catalyze its own formation. So here you start to think of how autocatalysis applies in a network. It's about the interaction of the system in establishing itself. It's not about a self-replication. And so this is also where I particularly agree with Stu, and Stu is not afraid of data and of thinking of ATP and DNA and stuff like that. So he says self-replication, homeostasis, and epigenesis may be the expected behavior of a particular class of matter. And I agree, I think so. An interesting thing of Stu's collective sets is that he showed they were probable, and then Mike Steele, when he formalized them in RAFs, showed that they are even more probable, most likely. So let's move to the implementation of the theory in RAF theory. This was done by Mike Steele and Wim Hoordijk, completely based on Stu's sets, and we've been working on it ever since, all of us together. A RAF is a very simple form to describe an autocatalytic network. It's basically you start with a food set, and you have a bunch of reactions that need to be catalyzed at some point, but you don't need to have the catalyst at first, as long as they can be produced at some point. One caveat that is really important, and you will see how important it is about RAFs, is that the catalysts can be in the food set. And not everyone that works on autocatalysis likes this, because traditionally autocatalysis means the catalyst is produced by the system, not in the food set. But life has catalysts in the food all the time. You know them very well. All life does. It's universal. Metals. Metals are catalysts for all life forms. And so when I started working with autocatalysis and the origins of life, I saw RAF theory as the best because it's very flexible as to how you study and predict the emergence of these networks. So I was not the first working with actual data with RAF theory. That was a study done by Felipe Souza in 2015 with a network of E. coli. So together, they had this very smart idea, the co-authors, that even though you have this self-referentiality between the code and enzymes, cofactors and metals are small molecules that exist, and they can catalyze reactions alone. This is in the literature. I have one example here with pyridoxal 5-phosphate. You may know it as vitamin B6. They can act alone very well in many cases. And 60% or more of mechanisms, enzymatic mechanisms, use cofactors or metals. And then you also have spontaneous reactions, which don't even need enzymes at all. So you can get rid of the complicated part and leave it for some smart person later and think, okay, enzymes are like Ferraris, but I can bike with just a small molecule. And I can also walk if there is an environment that allows my reaction to occur spontaneously. And so with this, they found that the E. coli network could have RAFs with small molecules in the food set. So what I did was apply this to the origins of life by parsing a huge amount of data. So we started with KEGG, which we heard about this morning. It's a database of biochemical reactions. I filtered the eukaryotes out because, as we know, they arose later. I think most of us agreed on that. Even that, some people disagree with. I also ditched the reactions that use oxygen because we agree, most of us, that oxygen arose later through cyanobacteria, which are very intricate prokaryotes. So I ended up in this network with 6,000 reactions of anaerobic prokaryotes. So, and then, of course, I had to get the small molecule annotation. So which cofactor, which metal can catalyze this reaction in theory? And this is the resulting network. It's a very large network, kind of like, you can't see anything here, but it's just to show you the size and more or less what we're working with as a canvas to look for autocatalysis. We did validate this network in terms of, as I said before in the literature, we knew that around 60% of mechanisms use at least one small molecule, and that's the result I got more or less. For 33% of the network, I could not annotate any small molecule. That means it's just a peptide that realizes the reaction. And then we had some spontaneous reactions as well. And of course, the network covers, of course, no, actually. The network covers all the enzyme classes that we know. So now I'm going to show you actual autocatalysis results. So I showed you the big canvas where we were going to look for autocatalysis, and now here are some results. So when we started with a very small food set, that's a list of compounds you see there, most inorganics that exist, we think, available on the early Earth, and the RAF was very small, just 8 reactions. Then we added to the food set some organic carbon that we now know is formed abiotically. Some very recent results show that, and we doubled the size of the network, which was interesting, but still very small. So we started to add organic catalysts to the food set. What this means is we don't know the way that they are or were produced, or if there were alternatives that could do their function. I showed you that table of the universal cofactors, which, by the way, was a completely different study before I even started, and then I ended up working with cofactors again, so they must be important. Anyways, so they each have a function, and it may be that in the early Earth, another molecule performed it. But what we show here is that when we add that possibility to the food set, the network expands vastly. And sorry, because there are some, but this is just to show you the size that the network grows very rapidly and very vastly when you introduce this possibility. So that's just the theory, right? That's like what's possible in the space of possibilities. But I wanted to do a bit more, and I wanted to actually check the networks of actual organisms. So we picked 2, one bacteria and one archaea, one acetogen and one methanogen that we think are the oldest for different reasons I can discuss with you later. And we crossed them. So basically, I found the RAF in each, the max RAF, and I crossed them. And this is probably the most interesting result of the paper, is that we did find a core network that was really interesting because it produced 6 amino acids and 3 of the nucleobases, acetyl-CoA, which is kind of the central hub of carbon metabolism and lipid. And another feature is that it had disconnected glucose metabolism, which other authors on early metabolism also agree with. So we continue to work with autocatalysis with Dan Hassan and Mike Steele. I'm helping develop this tool we call CatliNet. So if you're interested in autocatalysis and visualizing flux in autocatalytic networks, you can use this. It's very user-friendly. You just upload your data and ask, is there autocatalysis? I'm simplifying it, but you can email me and I'll help you. So I've been working on finding smaller networks because all my colleagues that do experiments say, Joana, that's too big. I cannot ever do that in the lab. So one of the small things we found is that you probably saw the jump if you had a close eye in that graph. When we add NAD or ATP, because they can interconvert, you gain both functionalities, and the network has a big expansion. So this is a reaction that me and collaborators are really interested in. Later with Stu, last year, I published this work that I basically just looked for RAFs in all of prokaryotes that are in KEGG, kind of as a proof of principle. We used 2 food sets, one that is very rich, the other one just with NAD. All of the networks I tested contain RAFs. 300 reactions appear in the majority of RAFs, and they are evolved in most of the categories of central metabolism. So this seems to be a very fundamental property of metabolic organization. The size of the RAFs grow linearly with the size of your genome. So as the more reactions you have, the RAF will grow concurrently. And this indicates that autocatalysis is independent of trophic mode. So no matter what you eat, what kind of prokaryote you are, autocatalysis seems to be very fundamental, at least in theory, computationally. So at the center, as I was talking about this interconversion between ATP and NAD, they are really interesting molecules because they are the universal, again, currencies of energy and redox power in life, and they can interconvert. And also, their moiety, adenine, is the first letter of DNA and RNA. So there must be something here about the origin of life as we know it. So I'm asking, is this a solution or a necessity in chemical evolution? I think we've been further from finding out. So one curious thing is I asked Joe Moran, a very accomplished prebiotic chemist that I collaborate with, do you think some of these molecules have been here since the origin of life? Because they don't degrade, they just pass from cell to cell. And he was like, you may be right, actually. So you may have some ATP and NAD in you that is here since the origin of life. So again, we're continuing to expand RAF-TIR. We just published this in bioRxiv. Why are we working so much on the theory? Because we're very data limited. The data set that I used hasn't grown. KEGG is kind of the same still, but we are expecting to get data soon through Dayhoff and other collaborators. So one thing that we're doing now with CatalyNet, you can identify strictly autocatalytic RAFs. That means all the catalysts have to be produced for those traditionalists that want their closed autocatalytic network. But again, that won't apply in the origins of life, I'm afraid. We're identifying all irreducible RAFs. That's like the smallest that you can go with the autocatalytic network. This can be a single reaction, as you saw in that example. And also something cool we're doing now is ordering reactions, so you can see what reaction has to come first for your RAF to grow to the size that you have. So just a little bit of incitement on some new ideas. I think I still have 5-10 minutes. From ponds to vents, from homeostasis to homeoresis, and from protocell to protofilms. So as you saw in the beginning of this talk, I was talking a lot about the environment in theories of life. I've been working very closely with people that study deep sea hydrothermal vents. And that's also not a chance factor. As a bioengineer, I always thought, wait a second. I've been designing reactors and trying to grow cells, and it doesn't seem like an open thing, especially with that high UV. This is very debated, by the way. You can try to debate me on it, but believe me, I'm used to it. Closed reactors are much better to grow pretty much whatever cell culture you want. But more than that, if you go deeper and you think about a soup or a closed pond, an open pond in equilibrium, and what life is, you kind of need a dynamic situation. You also heard about steady state this morning. You need a source and a sink. You need a continuous flow, a flow. So hydrothermal vents seem to be pretty much better as the environment for the origin of cells. at least in the perspective that I've built the past 10 years. But I'd love to continue discussing this, and I do with colleagues in Holland that still love ponds very much. So I told you I would talk about homeostasis and homeoresis. You know homeostasis very well. That's the maintenance of concentrations, of levels in the cell, temperature, and so on. For instance, here you have the cytoplasm levels being maintained compared to the extracellular. Homeoresis is what the protein is doing to keep that. So homeoresis is the set of dynamic processes that enable homeostasis. And I think it's a concept that is going to be very helpful also in the years to come when we think about the origins of life. So yeah, again, here you have, I like this quote by Leduc: the chain of life is a continuous one, from the mineral at one end to the most complicated organism at the other. We're all connected, even though we like to isolate ourselves a little. Just some recent results also on hydrothermal vents and why they're likely to be the origin, the environment for the origin of life. We did a thermodynamic study where we found that for the majority of the central reactions in metabolism, they are exergonic. They are likely to occur with hydrogen as a reductant instead of NADH. So thermodynamics seems to say, yes, now we need to find the kinetic conditions and the catalysts to make the network happen. So we're doing some flux variance analysis for this. We're also doing some kinetic modeling for this. Another idea that I want to bring you, because I want this talk to be mostly inspirational. If some of you wants to think about the origin of life, we used to think about protocells and the origin of life as like this individual unit that somehow has to arise. And lately I've been finding the idea of a biofilm much more helpful to think about these issues, because you need some growth and accumulation. Again, you probably need homeostasis first before you can go and leave the vent and do everything by yourself. So when you study biofilms, you see what keeps life together, basically. All these bonds that are keeping the polymers in the matrix together, they are the same that are inside the cell. So again, these are principles that apply universally to life. So a bit about the future. I think I'm fine with time, actually. I'm used to this now. Cool. So I think we need to think of a healthier community in origins. I don't know how much you know about the origin of life and origins of life research, but it's a very contentious field. And so I want to tell you a little bit about that and what I see scientifically for the future. So I don't know how many of you know Dennis Noble, but in systems theory, there is usually top-down and bottom-up approaches, but he proposed, to my knowledge, probably other people propose a different term, but something in the middle of top-down and bottom-up, which he called middle-out. I think it wasn't him, it was Sidney Banner that told him he should call it middle-out, what he was doing, modeling systems. Anyways, this is an outdated picture of how you can think of origins of life and artificial life and minimal life, outdated in terms of the content, but not outdated in terms of the idea behind it. So this was a review I wrote in my PhD that tells you a little bit about how you can position yourself and how you can navigate the space of systems biology when you're thinking about these concepts. So what do I see for the origin of life in the future? I think we will find out there is more necessity and determinism that we didn't know about, that the origin of variation before genes was probably through the origin of new catalysts and new redox and energy currencies. And then you will have, as Prigogine says, fluctuation and bifurcation that lead to emergence. So we're building this picture now where you start with a linear chemistry in the vents, and then you start to produce autocatalysts that enforce themselves. But we still need to think about structurally how these things could be stable enough to allow then for the code and polymerization, meaningful polymerization to emerge. So towards the code, I think we need to think more about necessary transporters or sensors. Membrane proteins are basically what says, this is me, this is not me. We need to think more about structures. So we're in a good point to do that with AlphaFold. And another thing we need to think about is at the subcellular level and in prokaryotes, things are very different in terms of evolution. I barely talked about evolution today, but we can do that. But neutral evolution dominates at subcellular level. So this is very important. Community. So we've been working hard in Olin since 2018 to build a healthier community where we can discuss and gain from each other's perspective. And so we published this paper in the conceptual realm, like trying to articulate heterogeneity in the field. It was a very strong collaborative effort that is paying some fruits now. And I'm very proud of this, to be honest. It's probably the proudest work I've done, more than autocatalytic networks that are really cool as well. So we just put this on bioRxiv, and this is our new paper of the network. And this is a mammoth of a review with 600 references on all of the techniques in the origin of life, analytical techniques, modeling techniques. So if you want to enter the field, now you have a compendium of how to do it, how to actually study life's origin. So I'm really proud. And we managed to put Olin as first author, so no one would be annoyed. And I'm just last author alphabetically because of the X. So you saw that image there on the left. This is a really cool artistic rendition that, together with these, I created with AI. I wasn't a big fan of GPT when it came out, of AI when it came out, but as many things in my life, I changed my mind, and now I'm completely changed. Not completely, let's say significantly changed, and I see the potential, and it's something we have to embrace. Otherwise, it's going to embrace us, and we have to start discussing it deeply. So at DayHoff, we're building models, AI models for the chemistry of life. I think in the next 5 years, we're going to be able to predict most of chemistry in aqueous solution. Maybe a crazy thing to say, but I think we're probably going to do that. Of course, I'm talking about simple chemistry, not polymers and complicated mineral surfaces. So yeah, let's start talking and thinking about AI. I'm not going to tell you about results there because we're basically building this, but we want to publish at DayHoff. We're not a company profit-oriented, we are science-oriented, mostly. And life is made of relations. Coming back to my friend Rosen, I think we've been thinking about life in the wrong way for a long time, about selfishness, and it's always a group, my friends. It's always a group of molecules, small, big, a group of cells. And so abstractions to units help explain selection, but models of groups reveal patterns of bottlenecks of emergence. So that's what we have to start doing if we want to really understand life. And the code, the black hole, the origin of true verticality. There are $10 million if you can solve it. Disclaimer, I'm a referee for this prize, and I've seen how far we are from solving it. I've seen a lot of good submissions, and unfortunately, yeah, so I'm just telling everyone, please think about this question because we need to think about it. It's a very hard question. And that's it. What a journey, huh? Thank you very much.
So thank you for this very inspiring talk. the time is open for questions.
The RAF networks expand, and does that mean that you get larger networks that are more interconnected, or that you get more and more modules? So is it like a very more fragile thing or a more redundant thing?
That's a good question. It's larger networks. I'm not looking about the modules here, and I think I wouldn't get much in terms of modules, but now that you're talking about it, I want to look there. But I think you need at least, I would say, 1,000 reactions to have some significant, at least as far as I know, metabolism. But I didn't look, and I will look now, and it's an easy thing to check, actually. Yeah.
Yeah, thank you. That was an extremely nice talk. For the origin of life, do you believe that to make an autocatalytic network, or the origin that this is something that is very likely to occur on its own, and there are many ways of doing this? Or do you think, like the origin of life, that this is an event with like a minuscule probability, and we're just extremely lucky that we're the only planet this is possible?
That's a big question, isn't it? I think it seems now that autocatalytic chemical networks, small chemical networks, meaning small molecules, they seem likely. There are also experimental results with autocatalytic networks of peptides and RNA. Now, there is a bifurcation moment that we're very stumbling around. That is the origin of the code. Or better said, the origin of encoding, genetic encoding. So the relation between genes and proteins, it may be that it requires a very specific, that's my suspicion, it requires a very specific environment. And we need to understand that relation to tell about the probability, to tell like how many times this environment happens in the universe, how many planets like this. We don't even know if it was on Earth. I mean, it's easier to study saying it was on Earth, but could have come from somewhere else. In fact, it was so fast compared to the origin of eukaryotes, say, that we understand much more about that origin process than about the origin of life. So I'm diverting, but it's hard to answer the probability question. And whenever I see people talking about the probability of life, it's like, we just don't know. You don't have data.
Maybe if we ever make it ourselves?
Yes, that would be the case, right? Yeah.
Thank you. Very, very inspiring talk, and I share many of your intuitions about this. Really, you briefly touched neutral evolution in just a line. Could you comment a bit more, explain a bit more about in what context you see that playing a big role?
I think in the context of not having genes that you are replicating very nicely in a very straightforward way. We don't know much of how chemical evolution happened, but we know there has to be variation and some form of selection. But now adaptation, it's harder to say. I can't imagine a complex system that is fragile arising and then having to adapt to something. The way I see it conceptually in my head is that it was one environment, and it has to be. You can have the origin of some complex molecules like adenine and then seeding that complex system, but I don't see a way for that system to adapt much, to have selection, adaptation and selection. Does that answer a little? It's not my major. Yeah, but I would love to hear your views on it. I would love to.
Are there questions from other rows on this? We're from the first row left. Please.
Just a very basic thing. I'm basically a computer scientist, so I couldn't follow most of the I'll be very interested in learning more. Are you going to make the slides available? I'll be very happy to work my way through them.
Yeah, sure. And
If
I
I
also
could.
heard this would be on YouTube. Someone told me to sign a paper.
They will be recorded, and you will find the whole lecture on
Yeah,
the
but I
YouTube
can
channel
also
of
share them.
ECLT. There is something which is called a YouTube channel of ECLT. So. Okay. Of course, now you are doing it in the opposite side.
Thank you for the great talk, very inspiring. It's a computer science talking, so I was wondering if you could comment on, you mostly talked about chemistry as a substrate for life, and I was wondering for us working in artificial life in computer simulations, what is your sort of thoughts or intuition about the substrate in which these properties for life could arise, for example, in a digital substrate?
It's a very intricate question for me because this is such a difficult topic to work in. And when I tried to abstract life, and of course I ended up with all the toy models of artificial life, and for me, until then, those didn't help much. So what I think you can do with computers is study properties of life. You can study replication, and studying homeostasis would be great. We need conceptual study of homeostasis. So you can grab Dyson's book and maybe catch some ideas from there or from the other authors I mentioned. So properties of life, like life is very dependent on matter and energy, life as we know it. If you want to call life something else, like if you want to say this model in the computer reproducing is alive, I don't support it, but I endorse it because I endorse all science. It's a matter of semantics. For me, what artificial life does great is advance our conceptual understanding of properties of life. So it would be fantastic if people would start to do that more for homeostasis and less for replication, because God knows how much replication models we've had. Not God, nature, whatever. You know what I mean? So like, I think starting to think of these other properties of life, not just replication, would be great, really great. I really hope this wasn't harsh.
No, please.
There are lots of people in Holland that work in artificial life, and we have very healthy conversation like this. It's just what life is for me, it's matter and energy.
And thank you for your emotional talk. This was really nice for me, and I don't know anything about chemistry because I am a kind of trained as a mathematician, and I may be an economist now of complex systems. So if I take, for instance, Rosen sentences, biology is concerned with the organization of relation, and they just change the biology. For instance, I say information is concerned with the organization of relation. Then I scratch a fundamental problem in economics and social sciences. We don't know a lot of things, and we don't know how interaction is producing something which is changing the nature of interaction itself, which looks like, I mean, in a weird space, it looks close to what you are looking at. So, I mean, this short introduction ended. Are you aware of works where similar ideas are applied to economics and social sciences, or can you kind of help me expand my imagination beyond the usual limits where information is a vector with numbers and so on and so forth? Do you have any thought of that? On that?
I have a little because of my close collaborator, Mike Steele. He's a mathematician, and he has applied RAFs in many other areas. So I'm his biochemistry origins person, but he has someone with psychology, someone with economy, someone with ecology. So if you go to Mike Steele's Scholar, you can Scholar page, Google Scholar, you can find his papers where he has worked in other fields with RAF theory. So I know that RAF theory has been applied in many other fields because it's so flexible. With regards to others applications in other fields, I don't know. I'm sorry. But that's a place to start. And Mike cites other people. So he will, yeah, he's a good person.
Okay.
About ecology, I was thinking, so we say that maybe there's a way for RAF sets to develop in prebiotic chemistry from geochemistry, okay? And then we don't know how to go on. Is there any work concerning RAF sets in which we consider how two different RAF sets may interact, like an ecology of different RAF sets, each with its own food source and waste?
I think there is one paper from Vim with Mike or not with Mike, but to be honest, I don't know exactly. I can check for you. But that's something we were discussing in Santa Fe last year. How cool would it be to see this? The problem is the data is so limited. We're so dependent on this keg, and it's great, but it's terrible, you know? And when you start to think about geochemistry, that's chemistry that's not in keg. Keg has chemistry of life, but not like the adjacent space, you know? And so, yeah, but I'll check that paper for you, remind me.
Okay, I think we should leave you free, but just one comment / question. When I see your work, I'm impressed, I mean, the one on the ancient metabolism. I'm impressed by the fact that they are small molecules. I came to know RAFs and all that stuff through polypeptides, models of macromolecules, Stu Kaufman, binary polymer model, and the like. So I always thought of large molecules. And now you say, no, they are small. What is the role of large molecules, if any?
We don't know enough about the principles of how peptides catalyze things. That's something we want to do in Dayhoff. So to predict computationally an autocatalytic network of peptides is very, very difficult because we don't have principles or annotations or any data. So there's the work of Gonan Ashkenazi, but it's just, that's the system he built and that's it. You know, so we don't, but I agree, we have to move there. Polymers have to enter the stage, and in fact, glad that you said that. In my view, the origin of coding was some form of collective autocatalysis between short nucleic acids and peptides. In fact, there is some evidence recently from Nick Lane's lab on some biophysical constraints in those interactions. So we have to go there to the autocatalysis of polymers, but we need more data.
Okay, good.
Thank you.
Thank you again.
Thank you.
Now let us come back to the contributed papers. Riccardo Righi was scheduled